Hi everyone, my name is Emily Schuler. I'll be presenting with Cal, Vincent, and Tara on our research proposal, Unraveling the Link, Nutrient Deficiencies, Metabolic Syndrome, and Epigenetic Aging. Our goal is to understand how modifiable factors like nutrient intake influence not just metabolic health, but also the rate at which we biologically age, potentially offering new insights into strategies for healthy aging and disease prevention. Before we dive deeper into this presentation, I'd like to clarify some technical terms that we'll be using frequently. Understanding these terms will help connect the dots as we explore the links between nutrient deficiencies, metabolic health, and aging. Metabolic syndrome, otherwise known as METS, refers to a group of conditions including obesity, high blood pressure, and high blood sugar. Those symptoms together, or conditions, raise the risk of heart disease, diabetes, and other health issues. Epigenetic aging, this is about how our genes change over time. It's measured through DNA markers and helps us determine how fast our body is biologically aging compared to chronologically aging. DNA methylation, think of it as a gene switch. It's a chemical change to our DNA that can turn genes on or off without altering the actual genetic code. C-reactive protein is a protein made by the liver in response to inflammation. High levels in the blood are a sign of inflammation throughout the body. Biomarkers, simply put, a biomarker like blood sugar or cholesterol levels help us measure health or disease status. Oxidative stress, this happens when there's an imbalance between harmful molecules, free radicals, and the body's ability to counteract their effects with antioxidants, leading to cellular damage. These terms will come up throughout the presentation, especially when we discuss the biological mechanisms linking nutrient status, inflammation, and aging. Our group is exploring the question, is there an association between nutrient deficiencies symptoms associated with METs and markers of epigenetic aging? This question stems from the recognition that nutritional status, particularly deficiencies in key nutrients like magnesium, vitamin C, and vitamin E, may not only influence metabolic health, but also the biological aging process at the molecular level. We're particularly interested in how these nutrient deficiencies might impact MET symptoms through their role in promoting or reducing inflammation. Additionally, we aim to investigate how these metabolic and inflammatory changes could be linked to epigenetic aging markers, which provide insights into how fast the body is biologically aging. As I mentioned before, METs is a cluster of conditions that includes obesity, insulin resistance, hypertension, and dyslipidemia. These conditions significantly increase the risk of cardiovascular diseases and type 2 diabetes. A key factor driving METs is chronic inflammation, which worsens metabolic dysfunction. One of the primary markers of systemic inflammation is C-reactive protein, or CRP, which is produced by the liver in response to inflammatory signals. Since inflammation plays a critical role in the development of METs, identifying modifiable dietary factors can reduce inflammation, and it's essential for early intervention and disease prevention. Certain nutrient deficiencies have been found to influence inflammation, particularly in people with METs. Magnesium helps suppress pro-inflammatory pathways, improves insulin sensitivity, and reduces oxidative stress. Vitamin E, a lipid-soluble antioxidant, protects cells from oxidative damage. Vitamin C is a water-soluble antioxidant and neutralizes reactive oxygen species, or ROS, and this reduces overall oxidative stress and inflammation. Deficiencies in these nutrients are associated with elevated CRP levels and higher systemic inflammation, contributing to the progression of METs. Research in the field of epigenetics has identified what we call epigenetic clocks. These are biomarkers based on DNA methylation patterns that can predict a person's biological age more accurately than their chronological age. The three key epigenetic clocks used in aging research are Horvath age. This estimates biological age across approximately 51 tissues. Pheno age, which incorporates clinical biomarkers like CRP and glucose to reflect an individual's health status. Grim age, which predicts mortality risk and lifespan based on DNA methylation surrogates for inflammation, smoking, and metabolic health markers. Both METs and nutrient-related inflammation appear to accelerate epigenetic aging. Inflammation, measured by biomarkers like CRP, can influence DNA methylation patterns, leading to dysregulation in gene expression, cellular aging, and increased risk of chronic diseases. This means that the health of our metabolic system and our nutritional status don't just affect short-term outcomes, they can fundamentally alter how fast we biologically age. This is a core of what our research aims to explore.
So this is our research plan and methodologies. Our research is guided by 4 key objectives. First, we aim to assess the association between specific nutrient deficiencies, such as magnesium, vitamin C, E, and D, and epigenetic aging markers. Second, we'll investigate whether C-reactive protein, an inflammatory marker, acts as a mediator between nutrient deficiencies and epigenetic aging. Third, we'll examine how MET-S components might modify these associations, potentially amplifying or reducing their effects. And finally, we aim to explore the interactions between BMI, inflammation, and nutrient status to understand potential confounding or mediating effects. For this study, we'll use data from the National Health and Nutrition Examination Survey, NHANES, from 2001 to 2002, which is a nationally representative data set of U.S. adults. We are able to use all of the variables from this study. The inclusion criteria is adults aged 18 years old or older, complete data on nutrient biomarkers, MET-S components, CRP, and epigenetic aging markers. The exclusion criteria are pregnant individuals, such as which alters metabolic and inflammatory markers, and participants with acute infections are avoided to allow for confounding inflammation from temporary illnesses and cases with missing data on key variables. Our analysis will focus on 4 main categories of variables. The independent variables are nutrient biomarkers like magnesium, vitamin C, vitamin E, vitamin D, and others. The dependent variables are epigenetic aging markers, including Horvath age, phenoage, and Grimm age, which measure biological age through DNA methylation patterns. The mediator is CRP, representing systemic inflammation, to assess if it explains the pathway from nutrient deficiencies to epigenetic aging. And covariates are factors such as age, sex, and socioeconomic status.
For this study, we used a combination of statistical analysis, programming, and data visualization tools to explore the relationships between nutrient deficiencies, metabolic syndrome, and epigenetic aging. To analyze our dataset, we relied on R. R was used for statistical modeling, hypotheses testing, and visualizations using ggplot2 package. These tools allowed us to efficiently process and analyze large-scale health data. We sourced our data from the National Health and Nutrition Examination Survey from 2001 to 2002, which provides extensive biomarker, dietary, and health data. To manage and organize our findings, we used Google Docs for collaboration and Excel for preliminary data cleaning and structuring. To effectively present our findings, we utilized various visualization techniques, which are box plots, to compare CRP and epigenetic aging markers across MET-S and non-MET-S groups, scatter plots for regression lines to show relationships between nutrient levels and aging markers, and potential density plots to explore interactions between inflammation, metabolic health, and epigenetics. By combining these computational and analytical tools, we ensured a rigorous and reproducible approach to investigating how diet and inflammation influence biological aging.
The expected outcome of our research and data is that higher intakes of select nutrients, magnesium, vitamin E, and vitamin C, will be linked to lower C-reactive protein levels, especially in those with metabolic syndrome.
So this is our week-by-week timeline for the research project. I'll just quickly go over each week. The first week will cover descriptive statistics and summary tables, which are covering the statistics for key variables. Then we'll stratify by the MET-S status and epigenetic aging quartiles, and we'll compare nutrient and epigenetic aging markers across the MET-S and non-MET-S groups using t-tests or Wilcoxon rank sum tests. Week 2 will be data visualization. We'll create histograms and density plots for the key variables, use box plots to compare distributions of nutrient biomarkers and epigenetic aging markers across the MET-S groups. We'll check for outliers using the QQ plots and use correlation heat maps to examine relationships between the nutrients inflammation, and epigenetic aging. In week 3, we'll be checking assumptions for statistical tests. We'll assess normality using the Shapiro-Wilk test, check homogeneity of variance for group comparisons, and assess multicollinearity in predictor variables using variance inflammation factor. In week 4, we'll cover correlation analysis using Pearson or Spearman correlations, and then create scatter plots with regression lines to visualize the relationships. We'll use regression analysis using multiple linear regression to examine the associations between the nutrient biomarkers and epigenetic aging markers, and assess the interaction effects between MET-S and nutrients, and visualize interactions using interaction plots. In week 5, we'll do the mediation analysis, checking CRP as the mediator. We'll use Barron and Kenny's steps for mediation and the mediation package for bootstrapped mediation analysis. And that should leave us plenty of time to check the data and write the project report and record the video presentation.
If our data has significant associations between the nutrients and inflammation, it would support the idea that dietary interventions can help reduce symptoms in MET-S. No significant associations would suggest further research is needed on dietary patterns and nutrient absorption.
And here are the references that we have used so far, and we thank you so much for watching.